Hello everyone. In today's discussion, we will see Chapter 15, Review and Assessment. Let's see the learning objectives. Explain the main points of the chapter. Solve the assessment questions that are given under each lesson. Students, you can find this lesson on your textbook pages 442 to 445. Well, let's get started with this short summary of Chapter 15, Lesson 1, Selective Breeding. Selective breeding is the process of modifying the characteristics of living things, especially to enhance one or more desirable traits. Through selective breeding, humans choose organisms with wanted characteristics to produce the next generation. This takes advantage of natural variation among organisms and passes wanted traits to offspring. Now let's see some examples of selective breeding. So the numerous breeds of dogs and varieties of crop plants and domestic animals are examples of selective breeding. Let's see what hybridization is. Hybridization crosses the similar individuals to bring together the best of both parents in the offspring. Inbreeding is the continued breeding of individuals with selected characteristics. It ensures that wanted traits are preserved, but can also result in defects being passed on. Mutations are the source of biological diversity. Breeders introduce mutations into populations to increase genetic variation. And biotechnology is the application of a technological process, invention, or method to living organisms. Selective breeding is one example of biotechnology. Now let's see factors that affect mutation. Radiation and chemicals can increase the mutation rate. Diverse bacterial strains have been bred from mutated lines. Drugs can prevent the separation of chromosomes during mitosis, leading to polyploidy in plants. Such plants may be larger or stronger than their diploid relatives. Now let's see the lesson summary of 15.2. Recombinant DNA is DNA that has been formed artificially by combining constituents from different organisms. So, genetic engineers can transfer a gene from one organism to another to achieve a goal. But first, individual genes must be identified and separated from DNA. Once scientists find a gene, they can use a technique called the polymerase chain reaction, or PCR, to make many copies. Heat separates the DNA into two strands. As the DNA cools, primers are added to opposite ends of the strands. DNA polymerase adds nucleotides between the primers, producing two complementary strands. The process can be repeated as many as needed. Recombinant DNA molecules contain DNA from two different sources. Recombinant DNA technology can change the genetic composition of living organisms. Plasmids are circular DNA molecules found in bacteria and yeasts. They are widely used by scientists studying recombinant DNA because DNA joined to a plasmid can be replicated. A genetic marker is a gene that is used to differentiate a cell that carries a recombinant plasmid from those that do not. Let's see what transgenic organisms are. Transgenic organisms contain genes from other species. They result from the insertion of recombinant DNA into the genome of the host organism. A clone is a member of a population of genetically identical cells. Now let's see the lesson summary of 15.3, application of genetic engineering. Let's see the application in agriculture and industry. Genetic engineers work to improve the products we get from plants and animals. Genetically modified crops may be more nutritious or higher yielding. They may be resistant to insects, diseases, or spoilage. Some can produce plastics. Genetically modified animals may produce more milk, have leaner meat, or contain higher levels of nutritious compounds. Now let's see the application of genetic engineering in health and medicine. Recombinant DNA studies are leading to advance in the prevention and treatment of disease. Examples include vitamin-rich rice, human proteins made in animals, animal models of human disease or for research, and bacteria that produce human insulin. Gene therapy is the process of changing a gene to treat a disorder. However, gene therapy is still an experimental and high-risk technique. Genetic testing can identify hundreds of inherited disorders. Not all genes are active in every cell. So, by applying the technology of DNA microarray, scientists can study thousands of genes at once to determine their activity level. Now let's see the application of genetic engineering in personal identification. DNA fingerprinting analyzes sections of DNA that may have little or no function, but that vary from one individual to another. DNA fingerprinting is used in forensics, which is the scientific study of crime scene evidence, to identify criminals. It is also used to identify the biological father when paternity is in question. Now, let's see the lesson summary of 15.4, ethics and impacts of biotechnology. Let's see the profits and privacy. Most of the research in genetic engineering is done by private companies. They patent their findings and inventions to protect their investment and make a profit. The patents block other scientists from pursuing certain lines of research. To avoid such kind of problems, in 2007, the Genetic Information Non-Discrimination Act was signed in law in the United States. It prohibits discrimination based on genetic information. Let's see the safety of transgenics. There is controversy about the safety of GM foods. Proponents of genetically modified foods argue that GM crops are better, safer, and higher yielding than controversial crops. GM crops require less land and energy to grow, and insecticides need not to be applied to insect-resistant strains. On the other hand, opponents argue that the safety of GM crops has been neither adequately tested for long-term use nor regulated. Patents on GM seeds may force small farmers out of business. The resistance of GM plants to insects may harm beneficial insect species. Resistance to herbicides may result in the overuse of toxic chemicals. Well, now it's time to answer Chapter 15 assessment questions. Let's see some questions from 15.1. How do breeders produce genetic variations that are not found in nature? This is question number 4. The answer is To produce organisms with new traits, breeders can induce mutation, usually in bacteria, using radiation or chemicals, or they can create polyploid plants. Let's see question number 5. What is polyploidy? When is this condition useful? This is a very important question. The answer is polyploidy describes an organism with multiple sets of chromosomes. It can be useful to produce new species of plants that are larger and stronger than their diploid relatives. Now let's see some questions from 15.2 selective breeding. Question number 13. Describe what happens during a polymerase chain reaction. Another very important question. The answer is the first step is to heat a piece of DNA which separates its two strands. Then, as the DNA cools, primers bind to the single strands. Next, DNA polymerase starts copying the region between the primers. These copies can serve as templates to make still more copies. Question number 14. Explain what genetic markers are and describe how scientists use them. A genetic marker is a gene that makes it possible to distinguish bacteria that carry a plasmid from those that do not. Genetic markers are inserted into plasmids so that scientists can identify cells that have been transformed. How does a transgenic plant differ from a hybrid plant? A transgenic plant contains genes from other species, while a hybrid plant contains genes from only the parent plants. Now let's go to chapter 15, lesson 3, selective breeding questions. Number 21. Describe how a DNA microarray might be used to distinguish normal cells from cancer cells. Here is the answer. DNA microarrays allow scientists to compare the gene expression pattern of different cells. This kind of comparison can be used to distinguish cancer cells from normal cells. Let's another question. Describe 2 important uses for DNA fingerprinting. This is a very important question. Let's see the answer. DNA fingerprinting can be used to help solve crimes by matching DNA evidence from a crime scene with possible suspects. It can also be used to establish familial relationships, such as in cases of disputed paternity, by finding alleles in a child that are not carried by the mother and then matching them to the father's fingerprint. Well, now let's see some more questions from chapter 15, lesson 4. Question number 26. Give an example of a disadvantage associated with patenting genes. Here is the answer. Sometimes patent holders demand high fees that block other scientists from exploring lines of research. Question number 27. What is one argument used by critics of genetically modified foods? Here is the sample answer. Expensive GM seeds may force small farmers out of business, especially in the developing world. Students, this is the end of this discussion. See you in the next discussion. Bye bye.